development of human brain and the spinal cord. That is the topic for the today's session. Learning objectives. The student should be able to understand the origin of nervous system, primary and secondary brain vesicles, development of spinal cord, development of brain stem, and development of cerebral hemisphere. These are certain learning objectives. So the student should be able to understand all these things. Origin of nervous system. Nervous system develops from neural plate, a thickened slipper-shaped area of ectoderm. Thickened slipper-shaped area of ectoderm. Neural plate forms the following neural tube and neural crest. Neural tube that differentiate into brain and spinal cord. Neural crest give rise to autonomic nervous system, most of peripheral nervous system and ganglia. That is the origin of nervous system. Look at the image here. That is the anterior neuropore, first pharyngeal groove, second pharyngeal groove, second pharyngeal arch, first pharyngeal arch, pericardial elevation, first somoid, second, third, and so on. And ventral view shows like this. From neural plate to neural tube, how neural tube is going to be formed? The neural plate on dorsal side of embryo. Look at the image, dorsal side of the embryo. B, C, and D. B, C, and D. The plates fold to form a groove that is transformed into the neural tube. Look at the image. This one is the neural plate. At the center, it forms the groove. Here, more prominent. And finally, tube is going to be formed. Into neural tube, neural crest cells are pinched off from the neural tube and develops into sensory ganglion cell. Postganglionic autonomic neurons and peripheral glial Schwann cells, they comes from the neural plate cells. Look at the neural crest cells. They are giving rise these things. So neural tube, then neural fold, is neural groove, and finally neural tube that is come into existence. Primary plate, primary vesicle and secondary vesicle. Three primary vesicles. Forebrain at the uppermost part, cranial part, then the midbrain and hindbrain. Hindbrain going to be connected with the lower caudal portion of the spinal cord. So these three vesicles, primary vesicles, they are known as primary vesicle. They give rise 5 secondary vesicles. 5 secondary vesicles, telencephalon and diencephalon, then the mesencephalon and metencephalon and melencephalon. And melencephalon caudally connected with the spinal cord. So these are the 5 secondary brain vesicles. This portion from telencephalon give rise the forebrain, mesencephalon, midbrain, and the metencephalon to melencephalon give the hindbrain. Look at the image, 1A, 1B, then 2, 3A, 3B, 5 secondary vesicles. Neural tube, Cranial to fourth pair of somites develop into brain. Neural tube, caudal to fourth pair of somites form the spinal cord. So cranial most four somites give rise the tube brain, and caudal to fourth pair of somites give rise the spinal cord. Three brain vesicles form the cranial region: forebrain, midbrain, hindbrain. Forebrain is called as prosencephalon, midbrain mesencephalon, and hindbrain is called as rhombencephalon. Prosencephalon, mesencephalon, rhombencephalon. Forebrain, midbrain, and hindbrain. Look at the table. 3 vesicle stage, 5 vesicle stage. 3 vesicle stage: forebrain, prosencephalon, midbrain, mesencephalon, hindbrain, rhombencephalon, and caudal part of neural tube. Then 5 vesicle stage: telencephalon, endbrain, and dancephalon. So telencephalon give rise mature derivatives. What are they? Cerebral cortex, basal ganglia, hippocampal formation, amygdala, olfactory bulb. Dancephalon give rise the thalamus, hypothalamus, then the subthalamus, epithalamus, retina, optic nerves, and tracts. Then the midbrain or mesencephalon give rise the midbrain, and hindbrain, rhombencephalon, divide into 2: metencephalon, afterbrain, and myelencephalon, medullary brain. So metencephalon give rise the pons and cerebellum, whereas myelencephalon give rise the medulla oblongata, and caudal part of neural tube form the spinal cord. So 3 vesicle stage, then it comes into the 5 vesicle stage, and these are the mature derivatives from these 5 vesicle stage. Development of the spinal cord. The spinal cord forms from the neural tube, caudal to fourth pair of somites. Caudal to fourth pair of somites. The lateral wall of neural tube thickens and reduces the size of neural canal until only a minute central canal of the spinal cord is present at 9 to 10th V. So lateral wall, they are going to be thickened, and they are minimize the spinal cord, spinal canal into the 9th to 10th week. Neuroepithelial cells give rise to ventricular zone, which forms all neurons and macroglial cell in the spinal cord. Marginal zone of neuroepithelial cells form the white matter of the spinal cord. Proliferation and differentiation of neuroepithelial cell in developing the spinal cord produces following: dorsal and ventral part. Dorsal part form the alar plate, ventral part form the basal plate. Alar plate, cell bodies in which form the dorsal gray columns. In transverse section appear as dorsal horn, whereas ventral part form the basal plate, cell bodies of which form the ventral and lateral gray columns. In transverse section appear as ventral and lateral horns respectively. So 2 portions, dorsal and ventral. Dorsal give rise the alar, and the ventral give rise the basal plate. Dorsal give rise the dorsal horn in transverse section, and basal plate give rise the ventral and lateral horns respectively. Look at the image. It's the transverse section. It's the floor plate, a central canal is limitus. Then the alar plate, this one alar plate on either sides, basal plate on the other sides. At the center you can find the floor plate, and at the top roof plate is a transverse section of the spinal cord. and brain. Myelencephalon. Caudal part of myelencephalon resembles the spinal cord both developmentally and structurally. The neuroblast in the alar plates of myelencephalon migrate into marginal zone and forms isolated area of gray matter, that is gracile nuclei medially and cuneate nuclei laterally. In the dorsal horn, you can find these two, dorsal gracile on middle side and cuneate on the lateral side. And ventral area of medulla contains the pyramid, which consists of corticospinal fibers descending from developing cerebral cortex. So that is the myelencephalon, which is going to be attached with the spinal cord, developmentally and structurally the same as of the spinal cord. Myelencephalon, rostral part of medulla houses the diamond-shaped cavity of fourth ventricle. From medial to lateral on each side, following nuclei are formed. General somatic efferent, for example, neurons of hypoglossal nerve, special visceral efferent, neuron innervating muscles derived from pharyngeal arches, general visceral efferent, some neurons of vagus and glossopharyngeal nerves. Some neurons in the alar plate migrate ventrally and form neurons in olivary nuclei. Coming to metencephalon, just little bit above of the second part of rhombencephalon, the walls of metencephalon form the pons and cerebellum. The cavity of metencephalon forms the superior part of fourth ventricle. The cerebellum develops from thickening of dorsal part of alar plates. Some neuroblasts in alar plates migrate to marginal zone and form cerebellar cortex. Other neuroblasts from these plates give rise to central nuclei, largest of which is dent nucleus. That is the metencephalon. Cells from alar plates also give rise to pontine nuclei, cochlear nuclei, vestibular nuclei, and sensory nucleus of trigeminal nerve. Mesencephalon form the midbrain. The neural canal narrows and becomes cerebral aqueduct. Neuroblast from alar plates of midbrain into tectum pertaining to the roof and aggregate to form four large group of neurons, the paired superior colliculus and paired inferior colliculus. Neuroblast from basal plate give rise to a group of neurons in tegmentum to the base that give rise to red nuclei, nuclei of oculomotor, trochlear nerves, and reticular nuclei. So they are coming from the mesencephalon. Substantia nigra also forms from the basal plate. In the end, cerebral peduncles becomes prominent. Look at the image. Early development of the brain. Midbrain flexure develops into region of mesencephalon and slight bend. Cervical flexure can be recognized at the junction of rhombencephalon and spinal cord. Third flexure, pontine flexure, develops between the metencephalon and myelencephalon, and brain now contains 5 parts of the same as B, showing the medial surface of right of the brain in an 11 mm embryo. This is structure, and the medial surface of the brain in 43 mm embryo, human embryo. The diencephalon can be subdivided into thalamus and hypothalamus. Look at the image. Development of cerebral hemisphere A and B. The telencephalon vesicle expand rapidly to cover the rest of the brain and posterior ventral part of vesicle curves down and forward to form the temporal lobe. The lateral surface of brain of 7-month-old fetus. The insular region is bounded by the frontal, frontoparietal, and temporal opercula. Some of the more pronounced grooves have appeared, and hemisphere can be divided into 4 lobes: the frontal, parietal, occipital, and temporal lobe, and the insula. Look at the image, how they are going to be divided and rapidly in all directions. It is going to be enlarged and form the cerebral hemisphere. Outlines of cerebral hemisphere at the 25, 38, 53, 68, and 96 mm of the embryo. These are the developing cerebral hemisphere, showing the considerable enlargement from the end of embryonic period proper at post-ovulatory week. The lowermost triangle represents the entire brain at 25 mm, and uppermost triangle showing the brain in situ at 31 mm and almost natural size. One at the diagram, how the cerebral hemisphere is going to be developed at various stages. Development of the corpus callosum and fornix. Median sagittal section in a 10-week embryo showing the commissural plate and its development into corpus callosum. B. Median section through the corpus callosum and diencephalon in adult human brain. C. 3D drawing illustrating the relationship between corpus callosum, fornix, and hippocampus. So all these you can see how the relationship in corpus callosum, fornix, and hippocampus is going to be made. Development of corpus striatum and internal capsule. A medial surface of brain in 43 mm embryo. The thalamus is developing into diencephalon and corpus striatum in the basal part of telencephalon. Part of ganglionic eminence can be seen through interventricular foramen. B and C, transverse sections through developing brain. How the brain is going to be developed. Thank you very much. That's the end of the current session. Any question? Okay, why not? You can get from my office. Okay, thank you very much. Good office.